Hi, welcome back to Exercise Metabolism. In this video we're going to go over creatine synthesis, its metabolism, and then also its degradation in skeletal muscle. Make sure to like this video and subscribe to the channel for future videos and notifications. In this picture right here I have a basic schematic of creatine synthesis. All right, so you can see kind of the basic players here. This is the kidney, step 1 occurs there. This is the liver, step 2 occurs there. In fact, the synthesis is only 2 steps. All right, so in the kidney we have an enzyme. Some people will call it arginine glycine transaminase. Most people will just say guanidinoacetate synthase. So arginine and glycine are going to be condensed, and we're going to get the molecule GAA, which is guanidinoacetate. All right. This occurs in the kidney. This does not occur in the skeletal muscle. So the initial step is in the kidney where the urea cycle is. All right. So once we have this guanidinoacetate, it's exported to the liver. All right. Once guanidinoacetate is taken up by the liver, it's methylated by an enzyme called guanidinoacetate N-methyltransferase. All right. The methyl group comes from SAM, acetylmethionine, and we get out obviously acetylmethiocysteine homocysteine, and our product is creatine. This occurs in the liver. So step 1 is in the kidney where the urea cycle is. That's why we have plenty of arginine here, because the urea cycle synthesizes arginine. And the second step, guanidinoacetate methyltransferase, is going to be in the liver. That synthesizes the creatine, and then the creatine is going to be exported into the blood. And it's going to be taken up by the skeletal muscles. Okay, so for the most part, creatine synthesis is actually going to occur in the ultimately the kidney and the liver, and then it's exported to feed the skeletal muscle where it's imported through transporters. All right, so once we have creatine in the muscle, what does it do? Well, let's go over some biochemistry here. All right, so this molecule right here, this is a schematic of adenosine triphosphate ATP. All right, now we hopefully know by now that this bond in particular, right there where my mouse is, that bond between the phosphorus and the oxygen, this gamma phosphate bond right here is very, very high energy. So if we were to hydrolyze this bond, in other words, we were to break ATP into inorganic phosphate and ADP, we're going to release a massive amount of energy. In fact, at equilibrium, the Gibbs free energy is negative 30.5 kilojoules per mole. That's a whopping exergonic reaction. We release a lot of free energy doing that. Okay, part of the reason for that is that this phosphate anion is resonance stabilized, and also we have in general an increase in entropy because we're breaking apart ATP into ADP and phosphate. All right. It turns out, however, that in creatine phosphate, and so what is creatine phosphate? We just synthesized creatine. Well, it turns out that there's another reaction that occurs in skeletal muscle where we create the high energy form of creatine, which is called phosphocreatine, or sometimes you'll hear it referred to as creatine phosphate. All right. So what is creatine? Creatine is what we call a phosphagen, and there's a lot of different phosphagens in different organisms. In humans, the phosphagen is creatine, and a phosphagen is a molecule that stores excess phosphates. Okay, you can see that ATP can transfer its phosphate to creatine, and notably right here to make creatine phosphate or phosphocreatine. Notice that the phosphate is now attached to a nitrogen in phosphocreatine. All right, this is a reversible reaction. So first of all, for the reaction of creatine kinase, all right, and this is an equilibrium reaction that's based on the concentrations of the individual species, particularly in this reaction right here, okay? So if you have a lot of ATP around, you can use the enzyme creatine kinase and phosphorylate creatine to make phosphocreatine, generating ADP, all right? It is actually at rest in the muscle cell's best interest to store a lot of the excess phosphate as phosphocreatine, okay? And we'll talk about that on the next slide. But for this slide, let's suppose that we have a lot of phosphocreatine stored up, all right? Well, it turns out that phosphocreatine is a source for a very high power output of energy for the skeletal muscle. All right, so let's look at this. For the reaction of creatine kinase in the exercise direction, because this is a reversible reaction, but in the exercise direction, we're going to run this up from phosphocreatine to creatine. What that's going to do is it's going to take phosphocreatine, transfer the phosphate back to ADP to regenerate ATP, and we'll get back creatine. Now what this serves to do is it serves to generate ATP very, very quickly. Now the question is, why would it be beneficial to store the phosphate from ATP on creatine? Why would it be beneficial to do that? Why couldn't we just store the phosphate as ATP as is? Well, it turns out that when we look at a little more detail in the electron transport chain, where the ATP synthase enzyme makes ATP from ADP, if we actually remove or lower the concentration of ATP by storing it as phosphocreatine, it actually forces the reaction of ATP synthase to go towards more ATP. So it really is an equilibrium-based thing. We want to lower the concentration of ATP so that we can make more ATP. And the way we lower the concentration without hydrolyzing it is to store it on creatine as phosphocreatine. Not to mention, high levels of ATP also tend to inhibit some glycolytic enzymes like PFK1 and then also Krebs cycle enzymes. So it's in our best interest in multiple ways to store that phosphate on creatine. Well, notice here the hydrolysis of phosphocreatine. If we just hydrolyze it, it actually is negative 43 kilojoules per mole. That's actually about 12.5 more kilojoules per mole released than from ATP hydrolysis. So if we look at the reaction in the exercise direction, going from phosphocreatine to creatine, so we get that quick ATP synthesis from phosphocreatine, it turns out the overall Gibbs free energy of that reaction is negative 12.5 kilojoules per mole. Okay, so it's actually spontaneous in the exercise direction. So we store that phosphocreatine, then very quickly at the onset of very high intensity exercise, we transfer that phosphate back to ADP to make ATP, and then we have quick ATP. Okay? And this reaction is termed the phosphagen system because creatine and phosphocreatine are the phosphagens. Okay, and also the fact that this delta G at equilibrium of creatine kinase is negative, that helps drive the spontaneity of this reaction is what helps to drive the power output that we see for the phosphagen system over glycolysis or oxidative phosphorylation. So this phosphagen system is the highest power output of any of the energy systems. All right, now let's look at this in the reverse direction. This is the rest or postprandial direction. This means we're not exercising, we're at rest, and we just had a meal. Well, if we just had a meal, okay, well, let's suppose we have a lot of creatine floating around. Well, if we have a meal, we're going to run glycolysis. We're going to run other pathways probably as well. We're going to generate a large amount of ATP. We saw that the exercise direction, since this is reversible, is negative 12.5 kilojoules per mole, so we don't really have to do the math, but since we're doing the reverse reaction, it has to be positive 12.5 kilojoules per mole. So if we're going from creatine to phosphocreatine, it's actually non-spontaneous, meaning we're going to have to alter the equilibrium in some way. This is the overall free energy right here, not at equilibrium. This is the expression for it. We'd have the delta G at equilibrium, which would be positive 12.5 kilojoules per mole, plus RT times the natural log of the reaction quotient, which its expression is right here. Now, to make this delta G overall negative, which would be required if we want to run from creatine to phosphocreatine, what we would have to do is increase the creatine concentration and the ATP concentration. Well, it turns out at rest, when we're not burning a lot of ATP and we're metabolizing the stuff we just ate, ATP concentration rises. Additionally, because we're going to be doing biosynthesis at that point, creatine concentration rises because of these reactions right here, and the muscle cell is going to be intaking creatine. So because the denominator here is rising, that's going to make this term overall more negative, and once it surpasses positive 12.5 kilojoules per mole, overall delta G is negative, and we can run from creatine to phosphocreatine. So the whole point of showing that is that if we want to load up with phosphocreatine, we have to increase the amount of ATP and creatine that we have to force the equilibrium to go towards phosphocreatine. So whereas when we were running this direction from phosphocreatine to creatine, that reaction is just spontaneous, which is actually why the phosphagen system is so efficient. But when we're going the reverse direction to creatine phosphate, or phosphocreatine, we have to stress the equilibrium in order to get it to phosphocreatine. Okay, so this reaction is Le Chatelier dependent. All right. Now, like I had mentioned before, this is just another look at it, phosphocreatine hydrolysis is going to be very exergonic, all right? When we hydrolyze this, it's about negative 43 kilojoules per mole, and understand that once we get rid of that phosphate off of here, that regenerates this resonance in this guanidino group. Notice we have resonance stabilization. So that's also part of why we have this very negative free energy for phosphocreatine hydrolysis that we saw in this slide and this slide. Okay. Another reason for that is versus an oxygen phosphate bond, a nitrogen phosphate bond is even weaker. It's a much weaker bond than an oxygen phosphate bond, so that also makes this a very spontaneous reaction. Okay. Now, another look at this without structures is we see that the phosphagen system is going to be most active with bursts of heavy activity, heavy exercise. So for the most part, when we talk about the phosphagen system, we're talking extremely high intensity. If you go outside right now and sprint as fast as you can down the street, the phosphagen system, on average, is going to be maximally active for about 10 seconds, maybe. Okay, so this is very short duration. This is not an efficient thing for maintaining long durations of exercise. So you're going to be able to do very, very high intensity exercise, sprinting, powerlifting. Okay, we go to the gym and do a lot of weightlifting, Olympic lifts. That's going to be the phosphagen system active there, okay? But it's only able to be maintained for a short period of time, and if you want to regenerate your phosphagen system, the stores of it, you're going to obviously need to intake a lot of creatine and food so you can get lots of ATP. And so that means that for aerobic exercise, so very low intensity, high duration, we're not going to be using the phosphagen system. That's going to be relying on oxidative phosphorylation. And once we get past, once we burn all of our stores of phosphocreatine, then we're going to be relying on anaerobic glycolysis for exercise. Okay?